Hello, my name is Marc André Sirard from the Department of Animal Science at Laval University. It's a pleasure for me to be with you today. I'd prefer to be face-to-face and see your faces, but we get used to this new normal. So I'll be happy to be able to answer your questions after the presentation. I will be able to talk live. First of all, let me introduce to you a project we have that has been going on for 3 years and for which we have results as for the heifers that were born recently. That was carried out by Catherine Chaput, a PhD student at the Department of Animal Science. And this morning, we are going to speak about the effect of deferring or delaying insemination of cows under metabolic stress from day 60 to day 120. What could be the result of functionally and economically? The problem we have in our farms, postpartum, one of the problems actually, is that it's an important decrease in the reproduction parameters. So cows don't get in heat. Heat doesn't last too long, and so overall, it's an increase in the number of inseminations per animal, which is costly and not funny for anyone. It's because we work doubly. What are the causes or the contributing factors? You heard about them. There are several of those causes. There is the intensified genetic selection. We have now a hyper-functional cows that are very productive, and consequently, their metabolism has been slightly altered. There can be uterine pathology, metritis, and contamination, so that the heat, the estrus doesn't work so much, negative energy balance, and metabolic disorders occur. We will take care about the negative energy balance. So cows lose weight. Among the metabolic postpartum disorders, that is the one that is the most influential on their reproduction parameters. This negative energy balance will affect the profile, and the reproductive system knows that the animal is hungry and adjusts accordingly to a certain extent. This is a reminder, that the energy needs are huge after postpartum because lactation is growing. At that time, the animal can never eat enough to meet their needs, so they will get thinner, and that's okay. That's part of what the mammals do, and they will give an additional energy effort during lactation because lactation requires a lot of energy. The adipose tissues or the fat are solicited and contribute to the energy production. And that leads to the production of ketonic bodies. Essentially, it's what we call BHB. It's an indicator that fat is released from the adipose tissue and put back in circulation. Actually, this is processed by the liver. They put back in the circulating blood to give to the cow enough energy to continue their milk production and continue to stay alive. We know that the negative energy balance could affect embryo quality. That's our hypothesis. And that could explain the fact that cows are less fertile between days 40 and 90. And unfortunately, that's the period during which veterinary recommends to inseminate the cows. And we realize that it's more and more difficult, and we'll try to explore what we can learn from that period so that the embryos are not so good or reproduction is not as efficient. Our objective, our research objectives are, first of all, to determine the effect of negative energy balance at day 60 at postpartum by measuring the embryonic development. We'll see this. You see that some animals lose more weight than others. They have a larger deficit. We'll try to compare with the cows that have not so much of a weight decrease. We'll produce embryos, and we'll look what is the effect on embryos. We'll look at the gene expression in embryos. We'll look at their DNA also. Can those conditions have repercussions in the medium or long term? And we do this to find potential markers for heifers. It tells us, okay, this heifer has been produced or in a negative energy balance, so it could have a different energy production. The little experimental design we followed cows postpartum. At 45 days, we took the BHP to be more precise. That allows us to have an immediate response. And then cows that have more than 0.9 in the blood, it's not the same in levels in the milk. It's comparable, but it's not the same numbers. So cows that are higher than 0.9 have high BHP, and they lose more weight. And then we look at hormones. We stimulate cows to have several embryos. We'll inseminate them with seed, and we get embryos in flushes, and we flush those embryos, and they will be used to two things. First of all, we will analyze them to see how they are, how they are programmed, and then we will transfer a few to get heifers when the seed is sexed, to have an idea about whether these embryos will create heifers with a phenotype or characteristics that are different. The BHB blood level between the two groups is clear. We made the groups based on that. So of course they are different as studies. To give you an idea of the variation, we took cows that were at about 1.5 compared to those that were at 0.6. So it gives a good contrast, a good difference between the two BHB levels. Those are cows in the same herd with similar milk productions, but some lose more weight than others, and that's the type of thing we want to compare. In terms of reproductive performance, two groups gave approximately the same number of embryos. However, the embryos of cows that have more BHB, we know they're hungry. We have embryos that are more at the morula state. So they're a bit late. It means that these cows ovulated a bit later. There's ovulation, then there's fecundation, and the timing starts, so these embryos are a bit delayed, not as advanced, more of them, and so they mutated later. That corresponds with postpartum cows that have metabolic problems. Very often they don't move ahead fast, and they calve 1 day later, and we were trying to understand that. Then in this experiment, we have now embryos to do some transcriptomic analysis. We look at the quantity of expression of genes. We look at 25,000 genes at the same time per group of embryos. That's huge, and that corresponds to an immediate response to the environment. The embryo will tell us in the way they express themselves. He will tell us whether it's different because their mother at the time of conception, the mother was hungry. So we'll have a response, a Polaroid response, a snapshot, and we'll look at the mediation of DNA in the embryo itself, because there could be side effects later on that we can't see immediately, that could emerge only later. And we'll look into the blood of heifers that were born. Can we find something that might have persisted after the gestation so that these heifers are a bit different? The analysis of blastocysts, I don't want to get into the details, and it's cellular biology here. What I'm trying to summarize here is the problems or the differences we see in the embryos of hungry cows are metabolic problems. We see that the embryo has less gene expression, he is less excited, he has difficulty to breathe, he has mitochondrial problems, it has difficulty creating energy, and it puts itself into a way. It's a way that would put the animal in an economy mode. With the scheme, you see the tissue, this is the membrane in green. We see all the factors that control the embryo metabolism. And what we can see is that the low energy that creates the great quantity of BHB gives signals to the cells to say, oh, stop oxygenating yourself a little bit, produce less protein, recycle your old protein, breathe a bit less, and stop burning all your glucose because you won't get enough because your mother is hungry. So that puts the embryo in an energy-saving mode. Not enough to kill the embryo. Embryo are still viable, but they're in energy keeping mode, energy saving mode. They are told the environment is low in energy, so you got to adapt. When we put it all together, we see that the signals given by the embryos is that they stop multiplying, they try to survive with low energy, and they change a little bit their breathing to make sure they try to survive through autophagy. They want to survive there. Interestingly, we noted a transcriptomic signature of embryos in the past when we make embryos by in vitro fecundation. When we compare very young heifers at 8 months instead of adult heifers, we see that very young heifers give the same signals as a cow that is hungry. So a heifer that is growing is in energy saving mode. It gives embryos that has the same feeling as a hungry cow, but it makes a lot of sense. It's interesting to see that. In the terms of epigenetics, we looked at 130,000 sites, which we can see, and we saw traces that will affect the embryo for the rest of its life. Out of 283, it's very light. It's 0.2% of the modification that could take place. So it's not a big change that will push them to fall down. It's lie down. It's very light. Where do these modifications occur? They don't occur anywhere. It's not in cancer. It's not in the lung or in the digestion. It's linked to the energy metabolism, a response to stress, and that confirms that the embryo programming could have consequences in the longer term. So the embryo is in energy saving mode, and their fetus making program will also be in a saving program. So if the mother is hungry, the embryo tells to itself, I will be hungry if I get out of there. So don't make too much tissue. Try to make as much energy to survive to the next generation. So it's programmed for evolution. An interesting phenomenon here, we noted that the end of the chromosomes was hypomethylated. What this means is that the cells divide a bit differently. And we noted that in cases of cancer, but also in studies we made previously when we create metabolic stress in embryos in an in vitro culture. That's, again, a confirmation that what we see in the full genome is an indicator that the embryo has a metabolic perspective that is pretty much in imbalance or deprogrammed. So for the genomics component, the conclusion is that a negative energy balance has a characteristic effect on the embryo because it gives a specific signature in terms of gene expression, which is immediate. It reacts immediately, the embryo reacts, and possibly a long-term epigenetic signature, which forces the embryo to save its energy, and that could create a different metabolic program in the heifers. If I look at heifers now, recently we had an analysis of the blood of those heifers which have gone through one lactation at least now, and we did the analysis of DNA in the cell, the white blood cells in the blood. That's the cells that we compared. We compared 4 heifers of mothers that were hungry compared to mothers that were not hungry. We would like to have 25. We only had 4 of each, so it's a very small sample. It's not very powerful, but statistically, we were able to get some sufficient statistical power to be able to distinguish a certain number of things. The first being that the distribution of the DNA modifications is not everywhere. We see here pink bubbles and blue bubbles. The not pink dots correspond to genes that correspond to about 5% of the genome, but here they make up approximately half of the changes are in the genes and not in the 95% that don't code for proteins. What this tells us is that, whoops, it could have an effect, and it's not a random effect. The modifications we observe in the heifers in blood are associated with coding genes, with functions. So it's not just random. There is a function associated with that. And if we try to associate the place, the locus of these genes that were modified, what types of genes those are, they're not random either. They target digestion, they target the lungs. Most of them target pathways, metabolic pathways that have gene expression. We're not in white cells, we're not in the mammary glands, so the white cells reflect that the animal is programmed slightly different in terms of the metabolic functions. So these 4 heifers are a bit different from the 4 heifers that where the mothers had less BHB in their blood. So we're less hungry. If we look in more detail, almost all components of metabolism are affected. Amino acids, CH, breathing, lipid metabolism, sugar metabolism. So it's really a general phenomenon. It's a signature that is very special, which tells us that in the DNA of those cows, there is something that is slightly different. However, will that create important phenotypic changes? We would need a lot more cows to be able to answer that question. I'm showing you here the 4 heifers in question, the 4 cows that were produced from those heifers in terms of milk production after 105 days, 305 days, sorry. With cows, we can't speak about any differences except that cows with a lower BHB have something like 1,000 kilos more of milk. We can't say whether it's significant. With 3 cows, it's really not enough, but some of these cases were called in both groups. The first group is a random event. It's a question of fertility. Some cows didn't even go to a sufficient level of production after 4 lactations. So there is a potential long-term effect that we should start understanding so as to measure in a better way the potential consequences of having earlier insemination on those cows which have a negative energy balance. With Edith Charbonneau, we did a study on the economic parameters with a student called Catherine Couture and other collaborators like Deborah Sanch. They went in the data from Valacta and they looked at whether we could associate the delayed for cows that were delayed from 60 to 100 days before being inseminated. Could we measure an impact on the profit or the quantity of milk produced, the profit that this will generate if we wait before inseminating the cow, so their lactation is lengthened or it's delayed? And we tried to encourage inseminate our cows normally at 60 days, but to avoid a period of time where the cow is less productive. It's normal, but now we will see that this is not necessarily a significant loss. So Edith and Catherine divided the animals into 3 groups based on their productivity, and they made an analysis, cost-benefit analysis on this delayed insemination. If we compare cows that were inseminated at 60 days versus those for which we waited 100 days, we see that there's a little gain, as we can see, is the dollars per cow per year. It's minimal, it's $17. Or what this tells us is that it's not negative. It's not an automatic loss when you delay the insemination. And that wasn't done only in high BHB cows. It was done on full herds. If we involve this only on high BHB cows, we suppose that it would be even more significant or more important in terms of gains for these animals. So if we put all this together, we can conclude economically that the voluntary waiting period for these cows with a high BHB level has no impact or only a minimal and positive impact on dairy farms' net income. Knowing that only a small proportion of the herd will be affected by high BHB and that the economic impact is minimal, why not adjust the voluntary waiting period depending on the energy deficit? That's the economic conclusion that is added to our biological conclusion, even though we don't yet have all the data we need. Right now, there is a project which is in development, or one project in production with Roger Q at McGill University. In this project, we're going to look at hundreds of thousands of cows. What is the effect of BHB in the mother's milk? Since we don't have take any blood sample, we'll take milk, and just before conception, before and after. What is the effect of a higher BHB on the daughter's milk production? And we'll put 4 heifers on both sides. This is not powerful enough. Then we'll have higher numbers. It was necessary to do the pilot project so that we can justify that we can do the large scale one. And the other one will document in a much more precise way with thousands and even hundreds of thousands of cows, what is the effect on the daughter? And then we can account for that and transform this in economic gain or loss, depending on the voluntary waiting time for insemination. There's a very interesting potential for this project as we advance in understanding. So in conclusion, we can say that delaying insemination of high producers seems to be biologically beneficial for cows with a residual energy deficit at 60 days postpartum, measured in the milk or in the blood. In the milk, the basic levels are easier to distinguish, but the measurement in the blood can be done. Several producers respond themselves in the blood. So this is for cows that are positive or higher than 0.9. You give them a chance by inseminating them later. We stop and let them lose more weight before trying to inseminate them. First of all, it might not take, and that way we give a chance to the mother to realize that there is enough free food for everyone to prepare heifers that will be better programmed with a better perspective about their own future. Then, economically speaking, delaying the insemination of high producers seems to be economically beneficial for the farm's net income. So what we thought we'd have to balance and the negative effects we thought we would have are finally positive. So everything supports the conclusion that we should treat hungry cows slightly differently so as to delay their reproduction and breeding, and the costs are pretty much and the future quality of their heifers. So I would like to thank the people who took part in this study. So Untitled Chapter, Catherine Couture, Edith Charbonneau, all the cooperators at Novalait and Novalait to identify, and Chris Ad for allowing us to carry out these experiments on the farms. And thanks to you for listening to me. And I hope I'll be able to discuss with you in the coming minutes virtually. Thank you.